We are proceeding with this class. When I was explaining the difference between DNA and RNA, there is something that I want to add, and it will be very important. Looking at this point of propagation. In propagation, what is the difference between DNA and RNA? We said DNA is self-replicating. And I give you an example of what? DNA replication. That in the case of meiosis, for you to have a new offspring, the reason why a baby look like the parents, because of what? Self-replication of DNA. To have a photocopy of a new DNA. Why this one is synthesized from RNA here is synthesized from what? DNA. Means any information you see in RNA, it is propagated or released from what? DNA. Then reactivity. Now, all this, when we have been talking since, I have not told you between DNA and RNA which one is more reactive. That is what I want to tell you now. We said the CH that is bound make DNA fairly stable at carbon position 2 because the oxygen has been removed. Since it's bounded to hydrogen and oxygen removed, it is fairly stable, means it is stable. And the small groove in the helix, that is the double structure. They say the small groove in the helix make it less substable to enzyme attack, means it is not exposed to enzyme attack. But in as much it is not exposed to enzymic attack by enzyme attack, it is prone to UV light, means ultraviolet damage. It is prone to ultraviolet damage because the groove covering it was a small groove. That is what we mean. But looking at the reactivity as the RNA, the OH that is bounded in the ribose make it reactive, means the RNA is more reactive than DNA. Why? Because of the OH group that is bounded. And the large group make it more open to attack by degradation of enzymes. Okay? It make it more open. It make it more reactive, and also it make it more prone to enzymatic attack because of what? The large group. So now we'll be talking about functions of DNA. Functions of DNA. Now I want you to listen carefully to these functions of DNA. If I ask you a question about functions of nucleic acid, you can also write the functions of DNA because DNA is part of what? Nucleic acid. One of the functions was storage genetic information. I very earlier told you the genetic information that is stored in the nucleus, form of in a DNA polynucleotide that is in a nucleus. This genetic information is what we are talking about. So the genetic information, everything about organism information, everything is stored in a DNA, in form of a DNA in the nucleus. Maintains growth and repair. As human beings keep growing, the cell need to divide. The old cell need to divide to form existing cell. Like in the case of mitosis, division of the organs. And that's why it have to happen. Division of the organ have to take place for a small baby to grow up to a full adult. And at that point of that growth, it's also maintaining repair. Okay? Like a cell that is damaged can also be repaired. And the one that can't damage will automatically commit a suicide to replace by the new one. That is a repair. Then 2, 3, control all cellular activity. Everything about the human body is controlled at the cellular level. Nothing in human that is out of control. All the information is present in form of a DNA in the nucleus, is control all the life of the organism. 4, contain protein codes. The protein code is in form of what? A codon. What we mean is this: when the DNA is transcribed to messenger RNA, the messenger RNA now finds its way out of the nucleus into the cytosol of the cell. Then in the cell there, the tRNA, which is the adapter molecule, will bring the amino acid, the correct amino acid, to insert, and then the correct polypeptide will be found in the ribosome, which is released into the Golgi apparatus for the what? Formation of proteins. So, final one, which is 5, is what? Ensure continuity of traits. In this case, you all know that you don't know your great-grandfather, but you resemble and you look like them, because your father might have inherited a trait, a character, or the shape of a nose, the shape of the hair from there, and you also inherited it from your father, because it is continuity of a trait. And then there's something we refer as what? A nitrogenous base, in which you have been told in the class, and then there are portion of it. So this human DNA contain, in every human being, the human DNA contain adenine 30%, thymine 30%, guanine 20%, cytosine 20%. Functions of RNA. I've earlier told you, if I say functions of nucleic acid, you can write the functions of DNA, and you can also write the function of RNA. But in this case, look at it now. One of the functions of RNA is what? The messenger RNA performs the function by transfer DNA from the nucleus into the messenger RNA and translate it into the ribosome for formation of what? Protein. That's what we mean. So the messenger RNA is the one that do the work of transferring the genetic information from DNA to the ribosome. Messenger RNA transfer what? Genetic information from what? The DNA in the nucleus to the what? Ribosome for formation of protein. M means messenger RNA. T means transfer RNA. That's what we call it, tRNA. Transfer RNA. The transfer RNA decode genetic code in messenger RNA. So at this point of messenger RNA, the transfer RNA will bring the correct amino acid and insert for the appropriate amino acid to be formed and be released out of the cell, which all this thing happen in form of what? Genetic code. R means ribosome. So we call it what? Ribosomal RNA. So it's the structural component of the ribosome. And listen carefully, there's something we call what? Central dogma, which is the control of gene expression. Means, listen carefully now, for you to be a human being and all your life activity on earth, for every organism, this is how it happens. So all that we have been saying since, this diagram can explain it to you just like at a fingertip. This is your DNA, this place is your DNA, and at this place we are having your 5' and at the 3', this is your DNA, and all your information is present here. So at this point, for replication to happen, it takes place at this point of the DNA. In this point, there's nothing like formation of protein, but replication takes place. So out of this place, double-stranded DNA can replicate to have a single-stranded DNA, and which if another photocopy will be found, and so on and so forth. That's what means gene on what? On DNA. But and at this point, it is still in the nucleus. So in this point, the messenger RNA can just be transferred from this nucleus and then to what? Transferred to the nucleus. In this place is transferring. Look at it. We have exo and intros. Exo and intros. This exo and intros, everything is in form of the message, the genetic code in form of a message, and the thing will keep flowing out of the nucleus here, from here down to here. It is still happening in the nucleus. Look at now, messenger RNA, it is still in the nucleus, but out of the nucleus, look at how it's now flowing to the cytosol. The cytosol is the jelly substance that is in the cell. in which organelle is attached to. That's what we call cytosol. The jelly component in the cell that organelles find its way attached to it. That's what we call cytosol. So now that the DNA has been changed to messenger RNA, it finds its way out of the nucleus into the cytosol. So this is the ribosomal subunit for the synthesis of protein. So at this place now, the messenger RNA now, look at it now. Transcriptional control, RNA processing control, then RNA transport control means it has been transferred from the nucleus into the what? The cytosol. Then at this point, we don't have transcription again. What we have here is what? Translation. Translation means the messenger RNA has now will be translated into protein. Look at it. So this is protein. So it is this protein that is released into the target organs of the body that bring about the continuity of life. This is how everything in human body happens. So the one that is not happening at the level of replication here, it is following this process for the synthesis of protein. If protein is formed, every human being is made up of protein. Cell is made up of protein. Nothing in the body that is not made up of protein. Everything revolves around the protein in the body. This is the process for the synthesis of protein. And if a new child is about to be born, this is the process of replication for a new baby to be born. So everything in this life revolves around the central dogma, which is the control of what? Gene expression. Listen carefully. That I thank you for giving me your attention. I want you to go over these online lectures. It will help you to understand better what did I say from the beginning. Now you might have understand the structure of DNA and RNA, the structure of nucleotides. I want you to go, we have 5 nitrogenous base, know the structure. And then we have structure of nucleotide, know the structure. And so on and so forth. If you have any question, you can come to my online YouTube channel and place your question there while I will do justice to it. Or we meet in class, you ask your question and I will explain to you better. Thank you so much for your attention. I remain Matthew Ocheleka Itodo.